I'm Svetlana Ilvorska, Associated Professor of Biochemistry Department. Our lecture about general principles of metabolism regulation, hormones. The homeostasis in an organism regulated with different systems on different levels. The highest level of regulation with nervous system, intermediate level with endocrine system. These systems regulate intracellular enzymes activity. Hormones are organic, biologically active compounds of different chemical nature that produced by endocrine glands enter directly into blood and regulate metabolism of compounds and functions on the organism level. Hormonoids or tissue hormones are compounds that are produced not in glands but in different tissues. They are responsible for regulation of metabolic processes on the local level. Some of them, for instance serotonin, enter blood and regulate processes on the organism level. Specific stimulation for hormone secretion are nervous impulses and concentration of compounds in blood passing through the endocrine glands. There are such endocrine glands as hypothalamus, pituitary, epiphysis, thymus, thyroid gland, parathyroid glands, Langerhans islets of pancreas, epinephrine or adrenal glands, and sex glands. According to chemical nature, hormones classified into 5 classes. First class, proteins. They are hormones of anterior pituitary except ACTH, insulin, parathyroid hormone. Second class, peptides. ACTH, calcitonin, glucagon, vasopressin, oxytocin, hormones of hypothalamus. Class number 3, derivatives of amino acids are catecholamines, thyroxine, triiodthyronine, hormones of epiphysis. Class number 4, steroid hormones are hormones of adrenal gland cortex and sex hormones. Class number 5 are derivatives of polyunsaturated fatty acids, prostaglandins. Hormones are secreted into the bloodstream. Hormones of protein and peptide nature secreted by exocytosis. Steroid hormones continuously penetrate the membrane and they are not accumulated in cells. Their concentration in blood determined by the speed of their synthesis. Protein and peptide nature hormones transported in blood in free state. Steroid hormones and hormones of thyroid gland bound with alpha globulins or albumins. Catecholamines in free state or bound with albumins, sulfates or glucuronic acid. Hormones reach the target organs. Cells of these organs have specific receptors for hormones. There are 2 groups of hormonal receptors. First of them placed on the surface of cell membrane. These are receptors for peptide and protein hormones, also prostaglandins. Formation of hormone receptor complex lead to formation of secondary messengers inside the cell, for example cyclic AMP, and as a result phosphorylation of intracellular enzymes and their activation. Second class of receptors placed inside the cell, in cytoplasm or nucleus. These receptors are receptors for steroid and thyroid hormone. After biochemical effects, hormones are released and metabolized. Hormones are inactivated mainly in liver. Inactive metabolites are excreted with urine. The final effects of hormones' action are: hormones change the permeability of cell membrane. Action on the allosteric centers affect the activity of enzymes. Affect the activity of enzymes through the messengers. Action on the genetic apparatus of cells. Now I will characterize all endocrine glands. First of them, hypothalamus. It's located in the base of forebrain between thalamus and pituitary gland. It collects information from another structures of brain and from blood flowing through hypothalamus. Hypothalamus produces three hormones of peptide nature for posterior lobe of pituitary. These hormones are antidiuretic hormone or vasopressin, oxytocin, and neurophysin. Hormones migrate along axons to posterior lobe of pituitary gland. Hypothalamus bound with anterior pituitary gland by the capillary network hypothalamic portal system. Releasing factors and statins reach the anterior pituitary via this system. Secretion of liberins and statins by hypothalamus is regulated by nervous impulses and by the change of concentration of hormones in blood, so-called feedback regulation. Releasing factors stimulate secretion of pituitary hormones. Statins are antagonists. They inhibit secretion of pituitary hormones. Releasing factors are somatoliberin, tyroliberin, corticoliberin, foliliberin, prolactoliberin, luteinizing hormone liberin, and melanoliberin. Statins are somatostatin, prolactostatin, and melanostatin. There are hormones of anterior, posterior, and intermediate lobes of pituitary gland. The most important, anterior lobe. It secretes tropic hormones. Tropic because these hormones stimulate function of peripheral endocrine glands. Somatotropic hormone or growth hormone is simple protein. It's secreted continuously during the whole life. Secretion stimulated by somatoliberin, inhibited by somatostatin. Main function of growth hormone, stimulation of somatic growth of an organism and tissues, particularly bones, cartilages, and muscles. Growth hormone acts directly and through the stimulation of formation of such polypeptides as somatomedins or insulin-like growth factors. These factors are synthesized in liver. Somatotropic hormone regulates metabolism of proteins, carbohydrates, and lipids. For protein metabolism, it promotes the entrance of amino acids into cells. It inhibits catabolism of proteins and amino acids. Also activates the synthesis of proteins and nucleic acids. For carbohydrates metabolism, it has anti-insulin action. It activates insulinase of liver, activates the exit of glucose from liver, inhibits the conversion of glucose into fats. For lipid metabolism, it stimulates the decomposition of lipids and also oxidation of fatty acids. In the inherited hypoplasia of pituitary gland, dwarfism is developed. For treatment, use growth hormone. Hyperproduction of growth hormone before puberty results in giantism. Hyperfunction of pituitary in adults results in acromegalia. It's unproportional intensive growth of particular body parts. This disease mostly caused by tumor of anterior pituitary gland. Adrenocorticotropic hormone is hormone of polypeptide nature. Secretion stimulated by corticoliberin, inhibited by level of cortisol in blood. This hormone controls the cortex of adrenal gland where cortisol is produced. It promotes the increase of cholesterol content in adrenal gland cortex and its conversion into corticosteroids. Also activates the passing of glucose into adrenal gland and pentosophosphate cycle. It has melanocyte-stimulating activity. ACTH hyperproduction causes Cushing disease that elevates cortisol level in blood. This disease is characterized by different symptoms such as obesity, particularly of trunk and face, sprained limbs, stria, proximal muscles weakness, hirsutism, insomnia, impotence, amenorrhea, infertility, heart diseases, hypertension, polyuria, hypokalemia, hyperglycemia, glucosuria, formation of kidney bones, depression, anxiety, hyperpigmentation of skin. Thyrotropic hormone is protein. Secretion of this hormone stimulated by tyroliberin. The speed of secretion regulated according to the feedback regulation by thyroid hormones level. It's necessary for normal functioning of thyroid gland. It promotes the accumulation of iodine in thyroid gland and its insertion into tyrosine. It stimulates the synthesis of iodine-containing hormones. Gonadotropic hormones are follicular-stimulating hormone and luteinizing hormone. Chemical nature of these hormones are proteins. Secretion of follicular-stimulating hormone stimulated by folliluberine. This hormone stimulates the function of follicles in women and spermatogenesis in men. Luteinizing hormone stimulates the function of follicles and conversion of follicle into the corpus luteum in women and secretion of testosterone in men. Prolactin is a protein. Secretion stimulated by prolactoluberine. Prolactin stimulates the function of mammary glands. It provides the body with sexual gratification. Stimulates the function of corpus luteum. Secretion of progesterone. Stimulates the growth of tissue of prostatic gland in men. It's responsible for mother instinct. Lipotropic hormone is a simple protein. Function: mobilization of lipids from the pore. It has melanocytostimulating function. It decreases level of calcium in blood. The intermediate lobe of pituitary gland secretes melanocytostimulating hormone. Chemical nature of this hormone: peptide. Functions of melanocytostimulating hormone: its stimulation of melaninogenesis and adaptation of vision in darkness. Vasopressin or antidiuretic hormone: its peptide. It's secreted from posterior lobe of pituitary gland. Antidiuretic hormone increases the reabsorption of water in kidneys. It also increases blood pressure. Insufficiency of this hormone causes development of diabetes insipidus that characterized by polyuria, low density of urine, and dehydration of an organism. Next hormone that's secreted from posterior pituitary is oxytocin. Chemical nature of this hormone: peptide. Functions of oxytocin: its stimulation of contraction of smooth muscles of uterus during labor. This hormone stimulates milk secretion. In medicine, it's used for labor stimulation, to stop hemorrhage after labor, and for stimulation of milk secretion. Epiphysis or pineal gland produces melatonin, adrenoglomerulotropin, and inhibitor of gonadotropin. Melatonin, formed from serotonin, it regulates the pigment metabolism, regulates sleep-wake processes, performs function as a neurotransmitter. Adrenoglomerulotropin stimulates secretion of mineralocorticoids in the adrenal gland cortex. Inhibitor of gonadotropin inhibits the synthesis of prolactin and cholesterol. Pancreas is the gland that has exocrine and endocrine parts. Endocrine: Langenkar's islets. In the islets distinguish alpha, beta, and delta cells. Alpha cells synthesize glucagon, beta cells insulin, delta cells somatostatin, epithelium of ducts lipocaine. Insulin is a zinc-containing protein. It formed from proinsulin by proteolysis. Synthesis of insulin regulated by glucose concentration level in blood. Other hormones such as somatostatin. Sympathetic and parasympathetic nervous systems. Insulin destroyed by insulinase in liver. Target cells for insulin are hepatocytes, myocytes, and adipocytes. Insufficiency of insulin synthesis leads to development of diabetes mellitus. Main function of insulin is regulation of carbohydrate metabolism. It increases transport of glucose into insulin-dependent tissues. It increases permeability of membranes for glucose. It activates utilization of glucose through activation of glycolysis. It activates tricarboxylic acid cycle, activates pentosophosphate cycle, glycogen synthesis, activates pyruvate and alpha-ketoglutarate dehydrogenase, inhibits gluconeogenesis and decomposition of glycogen. For protein metabolism, insulin increases the permeability of membranes for amino acids. It activates synthesis of proteins and nucleic acids, inhibits gluconeogenesis. For lipids metabolism, it activates the lipid synthesis and saving of fats. For mineral metabolism, it activates sodium-potassium ATPase. Glucagon is hormone of polypeptide nature. This hormone is antagonist of insulin. Synthesis activated in state of fasting. Glucagon activates the decomposition of glycogen in liver. It activates gluconeogenesis. It inhibits glycolysis, activates lipolysis. Somatostatin is hormone of peptide nature. This hormone inhibits secretion of insulin and glucagon. It inhibits secretion of somatotropin and thyrotropin. It inhibits secretion of local hormones in intestine. Functions of lipocaine is activation of phospholipid synthesis in liver. This hormone stimulates the action of lipotropic factors. It activates the oxidation of fatty acids in liver. Thyroid gland produces two types of hormones. First type, iodine-containing hormone, tetraiodthyronine and triiodthyronine. They are derivatives of tyrosine. Second one is calcitonin, hormone of peptide nature. Synthesis of iodine-containing hormones regulated by thyrotropic hormone, that in turn stimulated by thyroliberin. Iodine-containing hormones necessary for normal growth of an organism, for differentiation, sex and mental development. These hormones regulate the speed of metabolism. In physiological concentration, these hormones stimulate synthesis of proteins and nucleic acids. In the increased concentration, they activate the protein decomposition. For carbohydrate metabolism, they accelerate the absorption of carbohydrates in the intestine and activate the decomposition of glycogen. For lipids metabolism, iodine-containing hormones activate the exit of lipids from the pore, their decomposition and oxidation. For energetic metabolism, excessive concentration of thyroxine serves as uncoupler of respiration and phosphorylation. As a result, decreased ATP formation and increased the heat formation. Hyperfunction of gland is diffuse toxic goiter or thyrotoxicosis or Graves' disease. Goiter is hyperplasia of gland. Hyperfunction characterized by hypermetabolism, increased body temperature, muscles weakness, weight loss with good appetite, tremor, emotional lability, insomnia, acts of time. Hypofunction of thyroid gland occurs in the deficiency of iodine in water, salt, air. It characterized by decrease of metabolism, decrease of body temperature. Hypofunction in children causes cretinism, that characterized by inhibition of growth, unproportional body development, also disorders of mental development. Hypofunction in adults causes myxedema. Endemic goiter is disease that caused by deficiency of iodine in water, salt and air. Calcitonin, hormone that's synthesized by parafollicular cells of thyroid gland. This hormone affects the metabolism of calcium and phosphate. It promotes the transferring of calcium from blood into bones. It inhibits reabsorption of phosphate in kidneys, so it decreases the content of phosphate in blood due to excretion with urine. Increased secretion of calcitonin, characterized by hypocalcemia, hypophosphatemia, and hyperphosphaturia. Decreased secretion of calcitonin, characterized by hypercalcemia, hyperphosphatemia, but hypophosphaturia. Parathyroid glands produce parathyroid hormone, hormone of protein nature. This hormone regulates metabolism of calcium and phosphate. It promotes transfer of calcium from bones into blood, inhibits reabsorption of phosphate in kidneys, also stimulates the absorption of calcium in the intestine. Parathyroid hormone promotes formation of active form of vitamin D in kidneys. At hyperfunction of parathyroid gland, Recklinghausen's disease developed. This disease characterized by hypercalcemia, hypophosphatemia, hyperphosphaturia, osteoporosis, calcium accumulated in tissues. Hypofunction characterizes by hypocalcemia, hyperphosphatemia, hypophosphaturia, and tetany. In adrenal gland, distinguish two parts, cortex and medulla. Hormones of medullary part are catecholamines, epinephrine, norepinephrine, and dopa. Nature, derivatives of tyrosine. Excretion, regulated by sympathetic nervous system and brain cortex. Catecholamines are stress hormones. They promote contraction of vessels, increase the blood pressure, acceleration of pulse, also contraction of uterus muscles. Epinephrine relaxes the muscles of bronchi and intestine. On carbohydrate metabolism, epinephrine acts for activation of glycogen decomposition in liver and muscles. Also activates glycolysis, pentosophosphate cycle, tricarboxylic acid cycle, and tissue respiration. For protein metabolism, it accelerates the decomposition of proteins. Lipid metabolism, it activates lipase, mobilization of lipids, and their oxidation. Cortex of adrenal gland responsible for synthesis of corticosteroids. There are more than 50 corticosteroids. They are chemical nature steroids, derivatives of cholesterol. Main two groups of corticosteroids produced in adrenal cortex: glucocorticoids and mineralocorticoids. Glucocorticoids responsible for protein, carbohydrate, and lipids metabolism regulation. Mineralocorticoids regulate metabolism of minerals. Main representatives of glucocorticoids are corticosterone, cortisone, hydrocortisone. Their synthesis regulated by ACTH. These hormones transported combined with proteins. Half-life time till an hour. In the decomposition, 17 ketosteroids are formed. They excreted with urine. Diagnostic significance: index the function of cortex of epinephrine gland and testis. Glucocorticoids demonstrate anti-inflammatory, anti-allergic, anti-immune action, also adaptive effect. They maintain the blood pressure, also maintain the volume of extracellular liquid. In regulation of protein metabolism, glucocorticoids stimulate catabolic processes in connective, lymphoid, and muscle tissue, but they activate protein synthesis in liver. Glucocorticoids stimulate aminotransferases and synthesis of urea. As for regulation of carbohydrate metabolism, glucocorticoids increase the level of glucose in blood. They activate gluconeogenesis. They inhibit hexokinase, so inhibit glycolysis. In lipid metabolism, glucocorticoids activate lipolysis, activate the conversion of fatty acids into carbohydrates. The most important representative of mineralocorticoids is aldosterone. Excretion of aldosterone controlled by renin-angiotensin system. Functions of aldosterone: its activation of reabsorption of sodium, water and chloride in kidney canaliculi. It promotes the excretion of potassium ions via the kidneys, skin, and saliva. Addison disease or Brown's disease is insufficiency of adrenal gland cortex. It caused by injury of epinephrine gland cortex or insufficient production of ACTH. It characterized by low blood pressure, loss of weight, weakness, anorexia, also pigmentation. Hyperfunction of adrenal gland cortex characterized as Cushing syndrome. It caused by hyperplasia or tumor of adrenal gland cortex. For hyperproduction of aldosterone, specific such symptoms as hypokalemia, hypernatremia, hyperchloremia, hypervolemia, edema, and also hypertension development. Sex hormones synthesized in sex glands, placenta, cortex of adrenal gland. A little amount of female sex hormones formed in male organism. Little amount of male sex hormone synthesized in organism of female. Female sex hormones are estrogens and progesterone. Male sex hormones, androgens. Estrogens are hormones of steroid nature. Estradiol formed in follicles of ovary. Estrone and estriol formed in liver and placenta in the metabolism of estradiol. Their function is development of the female reproductive system organs, ability to fertility in reproductive period. Biochemical functions of estrogens are activation of anabolic reactions in tissues of reproductive organs, inhibition of exit of calcium from bones, so in menopause osteoporosis development. Progesterone, also hormone of steroid nature. It formed in corpus luteum, placenta, and adrenal gland cortex. Functions of progesterone: to prepare the endometrium of uterus to implantation of ovum, inhibition of uterus contraction during pregnancy, stimulation of growth of mammary gland. Testosterone, it's representative of androgens. It's hormone of steroid nature. It formed in interstitial cell of testis and excreted as 17 ketosteroids. Functions of testosterone: its activation of development of primary and secondary sex features, stimulation of spermatogenesis. Biochemical functions of testosterone: its stimulation of synthesis of nucleic acids, proteins, phospholipids. As a result, increased weight of skeletal muscles. Testosterone keeps the calcium and phosphate in an organism. Derivatives of androgens are active anabolics that widely used